Cool. All right, let's get started in our last bit of information. So if you guys remember in our previous lesson, what we looked at was this concept of population genetics. So looking at how genetics are affected in a big scale, looking at populations, looking at a whole species. Yeah, looking at that genetic variation. And in that genetic variation, or genetic diversity, I should say, the more genetic diversity I have, the better chances of survival for my species. We know that because it just means that if the environment changes, we have more variation, so we're more likely to survive any changes that can come at us. The other kind of big concept that we talked about was this idea of population versus species versus organism. Remember that species includes everyone in that grouping all around the world, whereas population is talking specifically of the species that live in that area, and organisms are that individual. When we have different populations of the same species, they might have different allele frequencies. So that just means in that particular makeup, they have certain alleles that are higher frequencies than others. Say, for example, when we're comparing European populations to Asian populations, European populations have a higher allele frequency for the blue eye gene than when you compare it against Asian populations. So that is an example of allele frequency and how population can influence that. But at the end of the day, Europeans and Asians were all part of the same species. We are all Homo sapiens, so we can still breed with each other. So do keep that in mind. Are we okay with that so far? So allele frequency is just looking at the percentage of the different alleles you have. So say the dominant allele is at 20% and the recessive allele is at 80%. So we just look at a population and we just count them up. How many have the dominant allele, how many have the recessive allele, and it's just a percentage. So that percentage can vary depending on the population. Also keep in mind that with allele frequency, it doesn't mean that the dominant allele is necessarily going to be in the higher percentage. You could have a dominant allele that is quite rare. Say, for example, Huntington's disease, which is an allele that causes that disorder. And that one's actually dominant. Now, what happens with time is that a population has a certain allele frequency. Excuse me? Populations could have different allele frequencies, and within that allele frequency, you have then individuals with different phenotypes. Some of those traits will be unfavorable, and if they are seriously unfavorable, it could prevent the organism from surviving. And if it doesn't survive, it doesn't pass on those alleles. So that's the end of the road for that trait. Other alleles are more favorable, so it means that the organism can survive and reproduce. If they're surviving and reproducing, they're passing on those alleles. Now, different things known as selective pressures will influence what alleles get selected for and which ones don't. There's things such as natural selection, which is the environment selecting the traits that are beneficial. There's artificial selection, which is when humans select the traits we want, and that's how we get a lot of our crops and our animals and domesticated, things like that. The last one I forgot to mention on here is mate selection. So maybe certain mates are looking for particular traits, like, for example, the peacocks. So that's mate selection. So it may not necessarily be anything that has to do with it being beneficial to the environment, other than just attracting other mates. So like a peacock with the big tail that's very colorful is more likely to attract the mate than one that doesn't have that big tail. And then if they survive and attract a mate, they can reproduce, they can pass on their alleles. If over time we have a lot of changes in those allele frequencies, what can eventually happen is the species can change so dramatically and evolve. Remember, species as a whole evolves, that we could actually get a new species from that. And when we get a new species from that, that is our speciation. Yeah, speciation is where that word comes from. So that was our kind of ending point up there. So, for example, apes and humans are two different species. We can no longer breed together and produce fertile offspring. but our relation is that we have a common ancestor, just like when you're thinking about grandparents. So we like share a grandparent. But because of our different evolutionary selective pressures and the natural selection, we then selected for different alleles. And with that, we eventually selected alleles that are so different that we then become different organisms. All right? This happens over many generations. It's not immediate. Are we okay with that so far? So that was recapping the last lesson. Yeah. All right. The next thing we need to think about is something called genetic drift. So in that example, we were talking about natural selection and artificial selection, mate selection. There's some sort of selective process, whereas genetic drift is just change that occurs randomly. So write that down. Yeah. Sorry, it's doing that thing. So random chance in this case. Due to random chance. So, like I said, artificial selection, natural selection, mate selection, all of that is specific. It's happening like for a reason, whereas genetic drift is just random chance. And there's two things that we think about when it comes to random chance. The first being that we have just random death, like just a dying out of our species or dying out of the population. Such as a natural disaster. The second example is if we have changes in the population. The thing to also remember about genetic drift is that it will have different impacts depending on the size of the population. So thinking about it logically, who do you think would be more affected by these random changes? Small populations or large populations? Small, because there's not as many there in order to take on that kind of change.
Yes. Let's say random death.
Random death, yes.
Oh man. Small.
Affected. All right. The first one I'm going to talk about is the change in populations, and then we'll backtrack to random death. Actually, random death will probably be easier to understand. Yeah. Go with random death first. Okay, random death is the first one. So, one of the ways that we can have random death is through something known as a bottleneck. So, for example, I have a beaker, or not a beaker, a flask filled with different beads. All of the beads in this flask represent the same population. And obviously, there's lots of variety here because we see that some of them are pearls, some of them are different colors, different shapes. So each bead represents an individual in my population. So we have my little flask here. And they're all living happily. With the bottleneck, it could be anything that causes a large death in our population. Say, for example, natural disaster. For example, a volcano explodes. With that volcano exploding, there's only a handful that will survive. So if I pour out those, these people are the ones that survived. These all died. They're gone. Now, these people are left to rebuild our population. So that is a bottleneck. And when we look at the genetic diversity in this population versus the original population, we see that it's far less. So we're going to be losing a lot of the genetic variation with that bottleneck occurring. So for example, when I'm looking at these guys, I still have the pearls, I still have the red color, I still have the yellow color, I still have the blue one, I still have the pinky one. But this next generation here, there's not any orange, there's not any purple, there's not any of these different shapes. So you see how that bottleneck has killed off a huge number of our population, leaving us with just a few to repopulate. And that's going to shift in a quite dramatic, fast fashion the gene pool. So are we okay with that so far as an idea? Bottleneck. So this one, bottleneck is just a sudden reduction in a population's size. Could be also the result of hunting. Humans are really good at that. We hunt and hunt and hunt, and we overhunt things, and then we just leave it so that way there's a couple of those species left. So with that sudden reduction in population size, there are fewer survivors, so only a small number of survivors to reproduce with each other. And since I have a smaller group of survivors that are reproducing, then I would have a decrease in my genetic diversity. And this loss of genetic diversity could hinder this population in the future because it may not be able to survive because it has less genetic diversity. A good example of that would be the cheetahs. So cheetahs have lost a lot of genetic diversity because of bottlenecking effects, the main one being hunting. So these were all my cheetahs. A lot of them got hunted. I only have a couple left, and that's all I have as my choices. Are you okay with that so far? All right, next thing that we then have to think about is our change in population. So our change in population happens for many different reasons. One of the key ones we are thinking about is migration. Now, have you guys heard the term migration before? What does migration mean? To move. Yeah. So this is just movement. So let's say, for example, I'm just going to grab some colors. So I have a population. We'll give these guys some pinks. All right, so for example, this is my original population over here. And with the migration, let's say a couple of those individuals move into the other population. So I have this population, I have this population. They're all still part of the same species, though. Number 2, changes in population. All right, so let's say, for example, this group of individuals decide to migrate and they join this population over here. So the larger group has had emigration, sorry, and this has had immigration. Another M in there. Sorry, immigration. Now, this is actually a really good thing for us to do because that can start introducing more genetic variation. So you see how these guys have now joined this group, and now this group actually now has some purple as a result. So that has increased the genetic variation in that example because now we're having some more things in there. And when I'm comparing these two groups, are these two groups now going to be more similar than they were before? Yeah. So between groups, they become more similar. All right. Cool. However, something else could happen with that migration. Some of that migration could result in the founder effect. So the founder effect is when we have these guys going to a new location. And this is the founder effect. So these guys have left, and with these guys leaving, the new population that forms is only purple and the orange. So now we can start selecting for things that are very different. And that's how new species can arise, because if they have a founder effect and that founder effect has different alleles, they can keep evolving and evolving and evolving, and then they then become different from the original population because they're now isolated from each other. This migration and founder effect could be the result of the physical movement of things, say for example from animals from one island to another, and then they get isolated and they evolve in a different way. It could also happen because of physical landmarks like a mountain that's separating the two species. Lots of different kind of geographic reasons why that can happen. New Zealand is a good example of the founder effect and why we have such unique animals in our islands, because we have become physically moved apart from the mainland, and thus there were different selective pressures for our animals. So yeah, we have lots of different animals. We have very unique animals. Very unique. Some big. We have a lot of flightless birds because in our environment in that new location, we didn't have a lot of land predators. We didn't have a lot of mammals. So the birds didn't need to be able to fly. So that's what we see there. Are we okay with that so far? So when we're thinking back to these notes, something that we talked about were how do we gain more genetic diversity? First way was through mutation, because that gives us new alleles. The second way that it can do that is through migration. So we can add alleles from other locations. So when we think about the actual, like in real life, the migration of humans, with all the immigration between the different lands and countries, we can start seeing that there's more and more, biracial people and things like that, because we have that migration and we have that mixing of the gene pool. And there was like a picture of what they think the future human's going to look like when we blend all of our different ethnicities together. Yeah. All right, keep going. So yeah, those are new ways that we can get some new populations. Some other things. Oh, good. All right. So when it comes to selection of our traits, there are 3 different things that can happen with the traits. So we're just going to write ways of selection. Yep. So we have a spectrum of different traits. Yes. Now, one way that our traits can get selected is known as stabilizing selection. So let's say I have my range of traits, and the one that becomes popular is the one that's in the middle. So with the population size, most of my population is the middle trait, so I move inward. So the middle trait is selected. And the extremes are selected against. A good example of this would be baby weight. So there's a range in what is the ideal baby weight, which is in the middle. If I was too small, my baby would be underdeveloped, it would be premature, so it would be more likely to die off. Why would I not want to have a really big baby? Something could happen during the childbirthing process. It could kill the mother, it could kill the baby. So that's why we wouldn't want a too small baby, we wouldn't want a too large baby, we want a baby that's roughly in the middle. And that's why it's selecting for that middle pressure. Does that make sense? So the example would be baby weight. So too small, it's premature. Too large, we have birth complications. So that's the first one. All right, second one is directional. So we select to one extreme. So when we have directional selection, we select to one extreme. An example of that would be giraffes. So their selective pressure was to get longer and longer necks. The giraffe with the middle neck and the giraffe with the short neck wouldn't be able to survive because they can't reach the tree leaves. Directional can also go in the other direction as well. You might be selecting for the smaller trait. It just means that we're trying to select something that's on the end of the spectrum. Okay with that so far? All right, the last one we have is disruptive. Disruptive is quite a weird one. Directional. So with disruptive, both extreme traits are selected for, and the middle isn't. The middle one isn't beneficial. Say, for example, I have rabbits. and maybe the two colors of the extreme are like black and white, and the middle color, or say, let's go brown and white. Brown can blend in with the ground, white can blend in with the snow, tan can't blend in with either, which is the in-between. So that's why it'll be selected against. Does that make sense? So like brown versus white versus tan. And that's bunnies. So this one can hide in snow. This one can hide in dirt. The tan one can hide in neither, so it dies. It's too easy to catch. So thus the disruption to one of the two extremes. All right, that's everything that I have for you guys. What questions do you have? From a look through the vocabulary. Keep going. Yeah, I'll bring it over. Or you can come grab it. Actually, wait, Klein's still writing stuff down. Can I move it? Directional and then stabilizing. So stabilizing is the one in the middle. So there's one of three ways you can go when you have this the shift in the genetics. We stabilize towards the middle, we push to one extreme, or we disrupt and get the two extremes. Right. Cool. I just want to double check to make sure you guys know all the vocabulary. Are you guys okay with bottleneck now? We've done bottleneck today. Founder effect we've talked about. It's that group that leaves, finds a new location. Are you okay with gene flow? So that's the movement of the genes. I think, let me double check it. Gene flow. Oh, excuse me. Gene flow is the transfer of genetic material from one population to another. So, let me go back to my paper. Oh no, you guys have it. Yeah, I can. Right. So this here, this going from there and joining that population, that's gene flow. Yeah, just the moving. Because you have to remember when the individuals move, they're carrying their genetic material with them. And then that can get passed on to the next generation. Where'd my list go? Here it is. All right, immigration we've done. Thymine, are you guys okay with? That's, or sorry, cytosine and thymine. Yeah. Chiasma, are you guys okay with that word? So when you have your crossing over like that, That's called your chiasma. Oopsie, that's not a very good drawing. Yeah, it's a section of crossing over. Yeah,
Yeah, that's chiasm. Okay.
that's the chiasma.
Chiasm.
That there. You guys okay with directional and stabilizing selections? That's what we talked about today. Centromere?
Yeah.
Yeah, it's just that bit here.
It's just central.
Yeah. All right, we're okay with genetic drift?
Yeah.
Genetic equilibrium?
No.
No? All right, let's look that one up. So genetic equilibrium is when the allele or genotype in a gene pool, the frequency does not change from generation to generation, so it stays stable. Yeah, so there's no changes. So that's just a stable gene pool. No changes between generations. All right, mate selection we talked about. It's like the peacocks. Adenine we know.
Yeah.
Chromatid.
That's a single chromosome.
Yeah, so when you have your duplicated chromosome, 1/2 of that's your chromatid. Locus? Okay, locus is the location of the gene. Semi-conservative? No? Let me show you that one. So semi-conservative replication is very common in DNA. What that means is that, here we go. We have the original template, and that original template is used to build the 2 new templates. Semi-conservative means that we have one of the templates are new and one of the templates are old. DNA templates, the DNA sequence. Yeah, so if something was, so semi-conservative here, we have the parent gene, or parent, let me zoom up. All right, oh no. All right, so in a semi-conservative model, we have the parent DNA, and after replication, what happens is that we have one strand that's from the parent and one strand that's new. In a conservative model, it would create a parent that's like the parent and then a brand new one. And then dispersive would be you just have random chopping around. So we use the semi-conservative model so that way we have a template to build our next strand of DNA from. And that's what we keep doing. We use one old strand to build the new strand, always using an old strand to build a new strand. It's just something to keep in mind. So here's another example of it. So you see, when we break our DNA in half, we have to replicate it, and we use the old strand as the template, and the new strand then forms. So it's basically we always have one strand that is always existing. All right, cool. That's everything that I have to teach you. So you guys are ready to basically do everything for bio. It's all the vocabulary words that we talked about. Oh, and then disruptive selection we've talked about. So y'all ready to go? That's bio.